now move on to our second talk. And please do stay after, by the way, if you have time. We'll do more talking and maybe look at some data too. So our second speaker is Hsiu Chuan Lin from Barbara Trutline's group, and looking at the NGN2 system along with morphogen dosing and combinatorial morphogen readouts in single cell. So thank you very much. I'll stop sharing and you can take over. Thanks for being here.
All right. So first of all, thanks, Carlo, for having me here. My name is Hsiu Chuan Lin, and I'm currently a postdoc at ETH Zurich, working with Professor Barbara Trutline. And it's my pleasure today to present our work in profiling and programming in vitro human neuronal diversity at single-cell resolution. So human cells can be engineered in vitro from iPS cells into diverse cell types, as well as 3D mini organs called organoids. And since the iPS cells can be directly reprogrammed from somatic cells of each individual, these in vitrally engineered human cells and tissues have enormous potential to serve as an avatar to model human health and diseases, to serve as a model system for drug screening and disease modeling. And these avatars are good in a way that they're genetically tractable. They allow time course measurements in development and disease progression, and they can also be grown in big batch for screening. So this all seems very promising, but one major question is that how similar are these avatars compared to their in vivo counterparts? So the single-cell transcriptomics could serve as a good tool here, since there are already quite extensive human cell and tissue reference atlas out there. So with this reference atlas, we're able to assess the quality of the engineered cells, such as the cell type composition, as well as identify any off-target lineages. And we can also directly compare the similarity of transcriptomes with their primary counterparts. So my project is centered on neurons induced from iPS cells. It is very well known that iPS cells can be directly induced into neurons by simply overexpress one transcription factor, NGN2 or neurogenin-2. And I'm especially fascinated by the system because the resulting neurons called NGN2R neurons in these presentations are induced with very high efficiency and immature way faster compared to normal development. So NGN2R neurons has been widely used to model disease and for screening, and it is claimed to be purely cortical excitatory neurons. But at that time, no one has really systematically assessed the identity of NGN2R neurons. So together with my two colleagues, Zhisong and Sebastian, we take the chance and use single-cell RNA sequencing to systematically assess NGN2R neuron identity. We found that NGN2R neurons are molecularly heterogeneous, as shown in the three fights here. And you can see expression of either CNS or PNS markers in different neuronal fights. And then we compared the similarity of NGN2R neuron clusters to those of primary reference atlas, and we found that NGN2R neurons are similar to both CNS and PNS neurons. This means that NGN2R neurons are confused, and they do not belong to any of the in vivo neuron subtypes. So this seems quite surprising to the people in the field because it is not as homogeneous and specific as what people would expect. But if you look at the expression pattern of NGN2 in development or in embryos, the heterogeneity of NGN2R neuron become less surprising because NGN2 is actually widely expressed across the nervous system. So with this, we wonder whether we can pattern NGN2R neurons using morphogen signals that patterns the neural tube to really guide NGN2R neuron into specific diverse subtypes with regional identity. So the morphogen we use here are morphogen involved in neural tube patterning. Here you can see in developing neural tube, and the anterior posterior axis is patterned mainly by the Wnt signaling, and which can be modulated by Wnt antagonist KIR and Wnt agonist KIR and Wnt antagonist XAV. And retinoic acid patterns the more hindbrain spinal cord area, while FGF8 is involved in the most anterior part of the forebrain and the mid-hindbrain boundary. Well, the dorsal ventral axis is patterned by BMP4 and sonic hedgehog, respectively. So we take advantage of the morphogen that I just described and set up a combinatorial patterning screen on top of NGN2R neurons. We start with iPS cells, induce NGN2 overexpression with doxycycline, followed by AP and then DV patterning. And at day 10, the resulting patterned NGN2R neurons, which include 192 morphogen gradient combinations, were harvested for single nuclear RNA sequencing using the high-throughput split-pole combinatorial barcoding platform. So together with my colleague Jasper Janssens, we start to analyze the results, and we are able to profile among these 192 samples 200,000 cells and identified 101 molecularly distinct clusters. And with the control here, you can already see that combinatorial patterning could expand the diversity of NGN2R neuron subtypes. And with this, you can see how each AP and DV morphogen contributes to this diversity. And we're able to identify where we observe this very interesting mirrored cell subtypes generated by dorsal and ventral morphogens. And we annotate the resulting cells based on comparison to primary reference atlas. And here are the results. So the pattern NGN2R neurons are annotated based on the division, the region, and the neuron type. And so you can see that we have both CNS and PNS neuron types in pattern NGN2R neurons. And in terms of regions, they're mapped to diverse regions, including the forebrain, midbrain, hindbrain, spinal cord, as well as sympathetic nervous system, enteric nervous system, trigeminal ganglia, and dorsal root ganglia. And for neuron type, we got a majority of glutamatergic neurons, which is in line with the known identity of NGN2R neurons. But with patterning, we're able to also get cholinergic and noradrenergic neurons. We wonder how morphogen combination contributes to this neuronal diversity. So what we do is that we want to know, we look into how neural identity shifts underlying morphogen combinations. And here I use the BMP4 and retinoic acid gradients combination as an example. And in the dot plot here, each column is a sample or a morphogen combination, while each row is an annotated cluster or a neuron subtypes with their identity, the division, the region, the neuron type labeled as sidebars. Each dot means the presence of a cluster or a neuron subtype in the sample, and the size of the dots reveal the proportion of this cluster in the sample. So the first column here is a control without any morphogen treatments. And these are the neuron subtypes that are present in control. You can see that they belong to a little bit more CNS and a mixture of glutamatergic and cholinergic neuronal fights. And here are the samples that are treated with different gradients of BMP4, still without retinoic acid. And you can see that there are acquisition of new identities compared to control, and their identity are mapped to more peripheral nervous system and change from more cholinergic, as shown in the green here, to more noradrenergic, as shown in the blue. And here, the concentration of BMP4 does not do much, since the composition looks quite similar between different BMP4 concentrations. While the first concentration of retinoic acid also didn't do much, as it is quite similar to control. But if we increase the concentration of retinoic acids, we can again see acquisition of new states. And if we add BMP4 on top, there's again new states acquired. And we wanted to know what are the underlying mechanisms, so we trained a random forest classifier to model the gene expression underlying morphogen response. And here, so I lay out the sample as the same order as previous, but then each row now is a gene in response to morphogen. And we can cluster these genes based on their expression pattern into gene programs. And I label only the transcription factor here, but you can already see as an example, there's higher expression or highly regulated Hox gene expression in response to higher retinoic acid concentrations. So we use we use this program as a basis to construct a gene regulatory network that are centered on morphogens. So here you can see the black ones is each morphogen that we use, while each node here is a regulon, meaning transcription factor and their target genes. and the size of the node, meaning the number of target genes of this specific transcription factor. We color the node based on the regional identity that have highest activity of this specific regulons. So with this, we can see that morphogen combination do activate distinct regulon to shape the neural diversity in pattern NG-into-I neurons. As an example, retinoic acids are linked with RARB and HuxB4 regulons, which are associated with more hindbrain fights, while BMP4 are linked with FOX2B and SMART regulon, which is associated with more PNS fight. We could dive deeper to look into the specificity of each regulons in each of the neuron clusters or neuron subtypes that we identified. And with here, each column is a neuron subtype, while each row is a regulon. And the heat map here shows the specificity of regulons. And with this, we're able to identify regulons that are more specific to noradrenergic fate or more cholinergic fate. And we can also identify regulons that are specific to midbrain and hindbrain cell identity. And these regulons could serve as a basis for future or further engineering of specific neuron subtypes. So with all these molecularly distinct neurons, we wanted to know whether these neurons are functional at all and whether they're functionally distinct. So we collaborate with Andreas Hilleman group in our institutes with the help of Manu, Philip, Maria, and Martha, where we try to assess the function of pattern NG-into-I neurons using electrophysiological measurement of their activity using high-density microelectroarrays. So we basically cultured neurons on top of these tiny electrodes and measure their activity. And with this, we're able to get, for example, the spike footprints, sort out spike with different forms, inferring different neurons, and we can perform exome tracing, and based on the amplitude and the amplitude of the signal and the time difference to infer action potential propagation, as well as the morphology of the neurons. And we can, of course, also measure the network activity of our culture. And here is an exome tracing of one of our neurons, and you can see how their action potential propagates as well as their morphology. And since these electrophysiological measurement is quantitative measurements with a resolution of single neurons, we can actually do multidimensional analysis on top of that. So we wonder what are the neurotransmitters that are underlying this neuronal activity. So we've performed pharmacological perturbation screening of small molecules for some of the neurotransmitter antagonists, including the mPAR receptor antagonist CNQX. You can see that CNQX treatments do lead to a reduced network activity in our neuron culture, but that the basal activity as well as the degree of reduction after CNQX treatments is quite different in each samples. We cluster the samples based on their activity upon CNQX treatments, and with this clear separation between samples, we are able to infer that our patterned NG-into-I neurons are molecularly distinct. So far, I've shown you results of patterning on top of NG-into-I neurons, but in development, patterning actually happens before neural differentiation. So we thus wonder what are the neuron subtypes we will get if we first pattern stem cells into diverse pre-patterned neural progenitors before NG-into induction. And we also wonder how these input states would affect the resulting neuron fight. So with this, we set up a pre-patterning screen. Pre-patterning means patterning happened before NG-into induction. In contrast to the what I showed you previously, this is called the post-patterning screen, where patterning happened after NG-into induction. So the pre-patterning screen include the patterning of iPS cells with AP and DV patterning to this pre-patterned neural stem cells and progenitors, and induce NG-into after that. And we collect in total 96 samples of the pre-patterned progenitors for bulk RNA sequencing, as well as pool them for single-cell multiomes with both single-cell RNA sequencing and ATAC to really know what are these input states. And for the resulting pattern engine-to-RNA neurons, we also harvest for single nuclear RNA sequencing. So here with pre-patterning, we're able to get quite diverse input states of neural progenitors with very diverse regional identity, as shown here. And we're especially interested in how this input states affect the chromatin landscape, especially for the engine-to motif. So what we do is that we filter out engine-to motifs based on their accessibility, and then identify potential engine-to target genes based on their proximity to these peaks, and analyze the resulting biological process that they're involved in. So what we see here is that for genes that are regulated or associated with an accessible engine-to motif, it's largely involved in neural functions for neural differentiation or neural maturation. This is not super surprising because engine-2 is known as a pioneering factor, which are able to just open up the chromatin by itself and then induce the resulting neuronal fates. What's more interesting is these accessible engine-to motif. They are involved, or the resulting genes are involved in patterning and regionalization. And many of these accessible motifs are even differentially accessible in different progenitors. So we wonder how engine-to interact with this differentially accessible chromatin landscape and affect resulting neuronal fates. And here are some of the schemes that could help us to think through the process. So if here's the control, right? If engine-to overexpress on top of iPS cells, we get a control engine-to neurons. But if engine-to could serve as a general pioneering factor, regardless of this pre-pattern progenitor identity, it will be able to generate similar neuron, similar to control if we start with different pre-pattern progenitors. If engine-to it's not as general as a pioneer factor, but it actually conforms to the identity of these pre-pattern progenitors, it will generate neurons with similar regional identity compared to the pre-pattern progenitors. Or if we got more wild, engine-to interact with chromatin landscape of progenitors and actively influence resulting neuronal fates, you will get something completely different. So we look into the resulting neurons, and we found that they have quite different regional identity and cell types compared to control. So we also try to compare the regional identity composition, or try to make a correlation of the regional identity composition in pre-pattern progenitors and corresponding neurons in each samples. And here each dot is a sample. And you can see that for this, we are able to see that the regional identity in each sample does not change much in pre-pattern progenitors and resulting neurons. This means that engine-2 is not actively involved in regionalization, but rather conform to whatever identity they are pre-established in pre-pattern progenitors. So we also wanted to know how are these pre-patterned neurons compared to those of the post-pattern neurons? Are they similar at all, or are there new identity acquired? So we integrate the data from pre and post-pattern engine-to neurons. And in this UMAP plot, you can see that these two data sets acquire very different UMAP, very different position on the UMAP, meaning that they're quite distinct. And we further analyzed the composition of their regional identity as well as cell type, which is not shown here. And we also found that they're distinct. And we wanted to know what are the gene regulation difference that drive this distinct fate in pre and post-pattern neurons. So we infer, again, the gene regulatory network. And if you look at the color of the edge, you can see that the morphogens can control like different regulons in pre and post patterned engine-to neurons. So lastly, we want to know whether combinatorial patterning could serve as a general strategy for cell subtype engineering. So we use the same strategy on top of induced GABAergic neurons from iPS cells by overexpressing ASCL1 and DX2, in contrast to the induced glutamatergic neuron by overexpressing engine-2. And with this additional data sets, we're able to, so with additional combinatorial patterning on induced GABAergic neurons, we're able to pattern or get quite diverse induced GABAergic neuron subtypes. Yes. And so in summary, we screen for neuron subtypes by transcription factor induced neurons plus combinatorial morphogen treatments with single-cell RNA sequencing readouts. In total, we profiled 700,000 cells in 480 conditions. We found that patterned NGN2I neurons are distinct neuron subtypes with diverse regional identity. And then combinatorial morphogen exposure could activate distinct regulons that potentially drive this neuronal diversity. We found, using electrophysiology, we found that pattern NGN2I neuron subtypes are functional and functionally distinct, and then change of input states could affect NGN2I neuron fights. Lastly, this combinatorial patterning could serve as a general strategy for cell subtype engineering. So with that, I would like to thank all the people who make this project possible, especially Jesper, Barbara, and Gray, as well as the electrophysiology team. I also want to thank Anne-Sophie, Gosha Wilco, and the whole quantitative developmental biology lab with all the support. And I want to also thank the Genomic Facility Basel for sequencing all my libraries. Thank you all for attention. I'll be happy to take any questions.
Thanks so much. Any questions? It's a huge amount of work.
I have a question. I was curious whether your patterns are validated by in vivo fetal human data sets or not.
Yes, we do this comparison, like here, I think. So for this annotation, we also include this this Brown et al. Atlas that recently came out. So yes, we do try to compare our pattern neurons, and this data set do contribute to our mapping.
So do you have a sort of an estimate to what extent your patterning reproduce what's
yeah. So yes. So I if I understand correctly, you want to know how similar are these neurons compared to primary, to sort of know whether it's reproducible of these in vivo neurons at all, or are they something different? So what I can say is that this is an in vitro 2D culture system. And what we observe is, so these annotation is based on comparison to control. If we compare a treatment to control, you can see the gene expression difference. And with this difference, you're able to infer whether they gain different regional identity, for example. But if you look at the absolute expression or absolute identity of these engineered neurons, they are not exactly reproducible of primary neurons. This is because these induced neurons share an induced neuron background, which make them less similar to primary. And in the future, it will be very interesting to erase this induced neuron background to make them more similar.
Can I ask a kind of provocative question to just keep us all thinking? So yes. I was thinking, you know, people want to make dopamine neurons for Parkinson's patients.
Yes.
So can I just call you up and you send me a protocol and I get lots of dopamine neurons? Doesn't really matter to me whether they have NGN2 or not, because after all, we treat Parkinson's patients with L-DOPA. We just need dopamine. So yes. Can you do that?
We do have some neurons that are mapped to dopaminergic neuron, especially derived from the induced GABAergic neuron datasets. So that would be a potential. But I would say that further characterization would be needed to really use these neurons for treatment.
So I
guess we could ask the same question to the previous speaker as well. Really.
Right, so Raya can respond because we actually measure dopamine and serotonin, so she can tell you.
Yeah, we perform basically proteomic analysis, and in the position from the
HPLC, not proteomic.
Yeah, HPLC for dopamine and serotonin, and basically in proportion with the cell types, the dopaminergic neurons, we see increased presence of these neurotransmitters. But again, of course, it needs to be, we could narrow down the concentration of the morphogen that should be needed to enrich and obtain a more pure population.
Well, I mean, I think the most remarkable thing was that it was in the posterior regions that you got this, and it was progressively decreasing as you went anterior. So it was not everywhere, right, in the device.
Yeah. And seems to be influenced by both morphogen that we tested. So
Right. Exactly. Yeah.
Yeah, I remember one of your UMAPs.
You know, when Lorenz first, let me just make another comment about dopamine neurons. So when Lorenz first started trying to transplant culture dopamine neurons, we tried to grow them from the primary tissue, right? And thinking we could grow neural precursors from other brain regions. But actually, that's very difficult to do. And there's a reason for that, which is that we've later discovered that the dopamine neurons are made by the floor plate. Now, the floor plate is very unusual because it doesn't proliferate much. So I think this is a very interesting topic, whether the point you just made, concluding, that we have to go on and characterize it more. Yeah, it's going to be very interesting to find out exactly how you can optimize for that particular class of neuron. I'm just raising it because there's so much interest in it and so much investment in it, actually. Thanks anyway, both of you. Wonderful.
There's one more in the chat for you, Swan Chen.
Yeah, so what is the combination of patterning factor that should be used to get pure CNS forebrain glutamatergic neurons? This we can see evidence. Sorry. So based on what we found here, we are actually able to get quite some forebrain glutamatergic neuron, especially in the pre-patterning conditions. So there are such conditions exist, and I can say that we're very close here. It's just that the thing is, in pre-patterning, the neurons I didn't bring up here. In pre-patterning, the neurons mature slower compared to those of post-patterning, so the maturation would take longer. This is presumably because of the epigenetic barrier that are pre-established in these neural progenitors compared to iPSCs. But yes, there's such condition exist.
Thank you for your presentation. I wanted to ask you, like maybe you said it in part, is like, is there for certain type of neurons like multiple morphogen paths to produce them, that you find different combinations of completely different morphogen can result in the presence of the same type of neurons?
Yeah, this is especially interesting. We are currently looking into that. We don't have a clear cut yet, but yes, our data would definitely be able to look into this. Thanks for the question.
Hi, can I ask a question? I'm not sure I understood well, but could you explain more about the difference between the pre-patterned neuron from the pre-patterned and versus post-patterned, right?
Yeah.
So, do you see some more diversity from pre-patterned neuron? They're actually you said that those are actually different, right?
Yeah, so they're different, and one of the major contributor of different is what I just mentioned. Their maturation status is very different. In post-parent neurons, they mature faster. In pre-parent neurons, they are more. They're still in the progenitor stage, at least at the day of our harvest at day 10. If we do this, we culture them for longer, they do become more mature. And specifically for other aspects of pre and post-parenting, I would say that since pre-parenting sort of follow the natural developmental path, it could make them more, like, at least in terms of annotation, it's way easier because of this in vitro induced neuron background is lower in these pre-parent neurons compared to the post-parent ones.
Mm-hmm. And so since you are actually triggering, you are using the patterning molecule, right?
Yes.
Do we see actually more regional diversity from the pre-patterned neuron?
So more regional diversity. So they have different proportion of regions For example, in pre-patterned conditions, there are more conditions that have higher proportion of forebrain neurons and hindbrain neurons. While in post-patterning, the regional specification, like in post-parent neurons within a condition, there are more regional identity present, meaning it's less specific. In pre-patterning, one sample has less regional identity, less regional identity, meaning that they're more specific. That's what we observed.
One more in the chat. Yep.
Yeah, they're still progenitor states. Doesn't mean that it's not effective anymore. Yes, so we actually look into that by we with these pre-patterned progenitors, we do a control without NGN2 induction at all, and to just really to look into the effect of NGN2, and NGN2 do still play a role to make neuron mature faster. If we don't do this NGN2 induction, their maturation status is way less compared to those with NGN2. So I would say that NGN2 is still quite effective in these conditions.